Hey everyone, welcome back to class. We are still talking about our cell membranes in this chapter, but we're moving a little bit away from what membranes are made of, how their chemistry kind of functions together, into what membranes are doing to let things in and out of the cell. So that selective permeability that we talked about in chapter 7, we're going to go a little more in depth here about how the cell allows material to come in or out of that cell membrane, or even an organelle, how it can allow things to come in and out through its membrane. So being able to overcome that permeability barrier is a crucial step of functioning for the cell. It needs to be able to move things in when it needs it and move things out when it needs it, but also to be selective about that. It doesn't want to move in waste, it wants to move in nutrients. And it doesn't want to move out the nutrients, it wants to move out the waste. So that selective permeability allows it to bring in the things that it wants to have in the cell and send out the things that the cell does not need for its proper functioning. So that involves what's called selective transport, or moving ions and organic molecules. And because membranes are able to selectively transport molecules, they are called selectively permeable. Selectively permeable tends to be the phrasing that I prefer to use, but you'll also see that phrase semi-permeable. So some things are able to come in, some things are able to go out, but it's not just a free-for-all, anything move anywhere. It is selective about what is allowed to move where. Here in this model cell, which is kind of a combination of plant, animal, generic stuff in it, but we see like here a lysosome is allowing hydrogen to come in, and it's going to break down this protein into amino acids. Those amino acids are allowed to go out. Here in the cell membrane, we're going to bring in amino acids to use to build proteins that this cell may need. We're going to here have a protein that is pumping sodium and potassium across it. This chloroplast is moving oxygen out as it is performing photosynthesis, bringing carbon dioxide in to perform photosynthesis. Between the chloroplast and the peroxisome, we have some molecules being exchanged between them. The peroxisome is letting out its products and bringing in serine to be able to oxidize it. The glyoxysome is bringing in fatty acids and letting out succinate. The nucleus is letting in the nucleotides, and it's sending out RNA, it's sending out proteins and bringing in proteins, those proteins that are going to help, turn on and turn off genes and regulate gene transcription. Here the cell has a protein that is allowing glucose to come in so that it can have energy to go to the mitochondria and turn into, waste products of carbon dioxide, bring in oxygen, release ATP. So all of these things need to come in and come out into the right place at the right time. Cells and cellular components will accumulate different substances depending on their different tasks. And what they're accumulating inside of themselves can have concentrations that are very different from their surroundings. So here the cell is bringing in oxygen and releasing carbon dioxide. It doesn't need to have those just equal to what's around it. It's going to want to bring in oxygen to be able to go through, electron transport and release carbon dioxide when it's broken down into sugar molecules. And it needs to be able to keep things in the correct concentration for the cell to work. And that's what homeostasis is that it's keeping its constant environment inside. Even if outside things are different, even if outside things are changing, we're going to keep that constant pH. Even if the environment that amoeba is in, maybe the lake is changing, it's still going to keep that homeostasis. That plant wants to keep its correct levels of water inside of the cell, even if the levels of water outside are changing. So that homeostasis is pretty important for a cell to be able to function. Most of the substances that are going to move across the membranes are going to be solutes, or they're going to be things that are dissolved. So dissolved gases, that would be things like oxygen, carbon dioxide, dissolved ions like the sodium and potassium, and small organic molecules like our amino acids or our sugar molecule. So those are all different solutes that get brought into and out of the cell. There's three quite different mechanisms that we're going to be discussing. So I'm going to kind of introduce them in this first video, and then we're going to talk about them in a little more detail. They are called simple diffusion, facilitated diffusion, and active transport. Some molecules are able to just come straight through this plasma membrane. That is simple diffusion. This is going to be dictated by their concentration gradient. So you can see it's moving from a lot to a little. Just like people on an elevator spread out, these molecules also want to spread out. And they say, hey, there's more space there. So they spread out to the other side of the membrane. However, to be able to go right through this membrane, you're going to have to be uncharged. You wouldn't be able to get through that hydrophobic core. You're going to need to be small to be able to fit through here. So there's very few things that can actually go through diffusion. You might be able to get oxygen and carbon dioxide through that way. But most things cannot just come through the membrane. Most things are going to require some sort of protein to enable that step to happen. So most transport is going to require a transport proteins. Transport proteins are going to be integral membrane proteins. That means they're bound into the membrane, and they go all the way through the membrane, making some sort of pathway for your solute to go through. Some of them are going to work passively. That means that they are still just moving down their concentration gradient. Things are spreading out, moving from where there's a lot to a little. So here there's a lot outside the cell, so it's moving in. If you had a lot inside the cell, it would move out. It's just going to spread out. That is what facilitated diffusion is. It's facilitated because it has to have some protein facilitate it or help it. But it's still diffusion. It's still moving from where there's a lot to a little. It's still not using any energy. The cell is not using up any ATP. Here you can see two different kinds of facilitated diffusion. This one's just kind of an open pathway that if it fits, it ships. Anything that can fit in there is going to go through there. This one is more of a selective kind of carrier protein that it has to be very specific. This is probably only going to move this little molecule, that certain molecule. Other things wouldn't be able to. So this is more of an open channel. This is more of a more regulated, specific protein pathway. Sometimes the cell wants to accumulate molecules inside the cell, regardless of what the concentration is outside, or wants to expel things from the cell, regardless of what the concentration is. So it's not going to just wait for molecules to spread out and come to an equilibrium across the membrane. Sometimes the cell is willing to use ATP energy to get molecules in. And that is what active transport is. Active transport is using a transport protein and moving things against the concentration gradient. So you can see it's not like there's just a little bit of these molecules here. It's going to selectively move these diamonds inside the cell, and it's selectively moving these circles outside of the cell against their gradient. So because it's moving against that concentration gradient, it is going to require energy. That energy is going to come from hydrolyzing ATP or breaking ATP down back to ADP and a phosphate group. Sometimes that energy can come from moving another molecule down its energy gradient. So you might have one thing moving in down its gradient to move something out without it across from its gradient. So here's a table. You can pause and go through this. Make sure you're very comfortable. Like you should be able to fill this out on your own. So simple diffusion is moving small molecules, water, small nonpolar molecules, oxygen, carbon dioxide, and other nonpolar things. They have to be specifically nonpolar. So oils and steroids. It is moving down its concentration gradient, so it is spreading out, coming to an equilibrium on both sides of the membrane. It does not require any energy. It does not have an intrinsic directionality, so it is not specifically only moving in or only moving out. It's going to move in whatever direction the concentration gradient calls for. It does not require membrane protein, and it does not have saturation kinetics or competitive inhibition, which we haven't talked about, but that means without a membrane protein, it really doesn't have any way to block a protein. Then facilitated diffusion is going to move still small polar molecules, so we can still move water. We can move large polar molecules, so things that are large and have a charge or just a hint of a charge that aren't able to go through diffusion. things that are polar like glucose, or things that are fully charged and ionic like sodium, potassium, and calcium, these are going to need some sort of channel. So they're going to go through facilitated diffusion. It is still moving down its concentration gradient, down its electrochemical gradient. So it's still moving from where you have a lot to where you have a little, to come to an equilibrium across that gradient, across that membrane. Because it's moving down the concentration gradient, it does not require any energy. It doesn't have any intrinsic directionality. It is going to go whichever direction will bring it to equilibrium. However, facilitated diffusion does require a membrane protein. And because it does use a membrane protein, that means that it is possible to have that membrane saturated or busy and always full. So there is saturation kinetics. And it does have competitive inhibition. You can block that protein and stop that transport. And then active transport is going to move large polar molecules and ions, so things that still require a molecule, but it will move them up their concentration gradient, up their electrochemical gradient. So you can accumulate things opposite, away from seeking equilibrium. Because we are moving against that desire to have equilibrium, it is going to take metabolic energy, and there's going to be an intrinsic directionality. The cell is specifically intentionally bringing it into the cell because it needs it in the cell, or it is intentionally moving it out of the cell because it wants it out of the cell. It's not going to reverse in other situations. So there is an intrinsic directionality to active transport. It does require a membrane protein still, and because of that, there is saturation kinetics. That means that membrane protein can be filled, and once it's filled, it's going to slow down, right? You're just going to end up with a line of people waiting to use the elevator to get into that cell. And you can competitively inhibit it. You can put some inhibitor that just clogs up that transport protein so that transport shuts down, or it inhibits transport. Okay, so we talked about that electrochemical potential a little bit in that table. So let's talk about that a little bit. When we are moving something without a net charge, the direction it's moving in passive transport is just determined by its concentration gradient. So if there's more outside and less inside, it moves inside. If there's more inside and less outside, it moves outside. So simple diffusion and facilitated diffusion are going to be exergonic movement. It's down the concentration gradient. That is sometimes referred to as a delta G. And when it's moving down the concentration gradient, it's losing potential energy, so it's a negative delta G. Active transport is going to be moving up the concentration gradient. So it's like you're climbing up the ladder. That's a positive delta G. So negative delta G is falling off the ladder, jumping off the ladder, doesn't take any energy. That's a negative delta G, moving down in potential energy. Positive delta G is moving up that concentration gradient or moving against its concentration gradient. However, that's only when it has no net charge. If an ion or a molecule has a charge to it, it's also going to be affected by its electrochemical potential. So that electrochemical potential means we're not only looking at the concentration of that molecule, we're also looking at the concentration of that charge. So if you have a small ion that's got a negative charge, and you've already got a lot of negative charges on one side of the membrane, it's going to be hard for that ion to move across the membrane. Even though it has a low concentration gradient, it's moving down its concentration gradient with the negative delta G, that electric charge is going to repel it away. So the active transport of ions across the membrane is going to create a charge gradient or a membrane potential across the membrane. That's the VM. That tells you how likely it is for an ion to move without even factoring in that concentration yet, just because it has that charge that is going to affect its movement. Most cells have a lot of negatively charged solids inside of them. And so that is going to mean that sodium is going to be brought inside. Cations like sodium are going to be brought inside, and anions like chlorine are going to be left outside. and that won't require as much energy. So in all organisms, active transport of ions across the plasma membrane will create that asymmetric distribution of ions inside and outside the cell. Okay, so we are going to finish up talking about one type of membrane that has been very well studied, and that is the erythrocyte plasma membrane. The erythrocyte is also called a red blood cell. These are the cells that take carbon dioxide and oxygen. They carry those through your body to deliver oxygen to your body, to take carbon dioxide as a waste to breathe out from your body. So these are some membrane kinetics that we've really studied, some of the best membrane proteins that are best understood. And the membrane potential is going to be maintained by active transport of potassium ions inward and sodium ions outward. And there will be these special pores or channels that allow water and ions to enter and leave the cell rapidly when they're needed. So we have here simple diffusion of carbon dioxide, oxygen, and water going on. We have facilitated diffusion, for example, using the GLUT1 transporter to move glucose into the cell. We have channel proteins, such as aquaporin, that are moving water in and out. And then we have active transport, maintaining our sodium and potassium energy kinetics that we need to keep that charge in and out of the cell. Okay, so we're going to come back and talk about all of those types of diffusion, describe them a little bit more, talk about our membrane proteins a little bit more, how they're helping that. So this hopefully gave you kind of a groundwork. Maybe it was a little bit more of a review of other classes, what you've learned in like your Bio 1 classes. But I'll see you back to finish talking about Chapter 8. Bye.